Welcome, AP Bio, to our Chapter 12 discussion on non-Mendelian genetics. So not all patterns of inheritance are going to be so simple as we've seen with just dominant and recessive. Sometimes things start to get sort of sticky. So we're going to explore that here. So the work done by Gregor Mendel was those really basic traits, and often they were traits that were only characterized by one gene. He's very lucky that he didn't go crazy trying to figure out more complex traits, and that the ones he stumbled on were very simple patterns of inheritance. But for a lot of traits, things are going to get more complex. So we're going to look at some of these things that get a little wonkier. So our first example is going to be incomplete dominance. And what incomplete dominance means is that our heterozygous pair, that's when the alleles are big and little, or they're different, are going to produce not the dominant trait, but a third form of the trait. So kind of an in-between stage. So you can see that with the flowers, where red is the dominant trait, white is the recessive trait, and then if you're heterozygous, you end up with a pink trait. And you can see that because what's happening is the recessive trait is being expressed, the dominant trait is being expressed, and since both those traits are in competition with one another, we end up with that blended pink. Another possibility is because you only have one dominant if the recessive isn't making a protein, like we talked about last time, that it's not producing as much red pigment, and so you see that pink version instead of that deep red, as if you were making two full alleles worth of red pigment protein. Similar to this is going to be the concept of codominance. And so this is where you see another form of blending where it's almost like they're fighting for space. And so codominance would be when both white and both red are visible. So that's like a Rowan cow where you see the white and red splotches instead of solid red and solid white. Really common form of codominance is in antigens on the surface of cells. So instead of showing one antigen or another, you see both antigens are going to be present. And we'll see an example of that here. Here's perhaps a great example of codominance in nature, combining ducks and beavers in order to make the friendly platypus. I should say, as a science teacher, if you cannot tell this is a joke, it is not how platypi are actually made in nature. Another form that we're going to look at is multiple alleles. So not everything is so simple that there's the dominant and the recessive allele. A lot of traits have multiple alleles that control for them. So for example, hair color, you may say like, well, there's not just like two types of hair color or even three with incomplete dominance. There's a lot of gradations there. And so when we do multiple alleles, like you can see here, we have the brown fur, the chinchilla, the Himalayan, and the albino. There's all sorts of different colors for these rabbits. There's more than just dominant and recessive. So what we're going to do is use capital and lowercase letters to still show dominant and recessive, because you'll find that some traits are dominant, some are recessive to some of those dominant traits. Sometimes there's codominance happening. And so we're going to use big letters for our, whether it's dominant or recessive. And then for the specific allele type, we're going to use exponent notation. So you can see here that there's like big C, there's the CH, the H, and then the little c representing that it's recessive. So like brown is dominant, and then there's all these different recessive types that have different alleles to them. A great example of combining these two, codominance with multiple alleles, is actually blood types. So there's those four major blood type groups, A, B, AB, and O. And there's also a positive, negative, but that's a whole separate gene. And this is a result of having three alleles. So there's the A allele, the B allele, and the O allele. And both A and B are dominant over O. And so that's why you see in the notation there, capital letter I is used for A and B, whereas it's a lowercase I for the O. because A is dominant over O, B is dominant over O. But A and B are co-dominant. So you can see if you have a genotype that's AB, you end up with AB blood. If you have AO or AA, you'll have A blood. BO, BB, B blood. And the only way to get that O allele or that O genotype or O phenotype is to have two recessive O alleles. And then when we put this into a Punnett square, just to show you an example, we're still going to use that exponent notation. So you have like A, O, B, O combined, you'll end up with A and a B gives us AB blood. B and an O combined gives us a B blood. A and an O blood gives us A blood. And then an O and an O gives us O blood. So these two parents, even though they're A and B, can give all four blood types, which is pretty neat. Here we see a big pedigree showing certain traits, and remember, when it's filled all the way in is if they have the trait, and if it's halfway filled in, they're a carrier. And do we notice anything bizarre or off about this? These are the old Russian czars and whether or not they had hemophilia. So, if you notice, only males have the trait. So what's going on with them? Hemophilia is an example of a sex-linked trait. And this was first found out in fruit flies, which we'll be doing a lab with fruit flies to see this. And these genes are carried on the X chromosome, not on the Y. Remember, the X chromosome is a lot bigger than the Y chromosome, and so there's a lot more DNA there, and so there's going to be genes that are on that X chromosome that don't have a counterpart on the Y. So if you're a male, you could just have one allele and nothing to match it. So with females, there's the possibility of being XX dominant, XX heterozygous, or XX recessive. But males, because they only carry it on the X, can either be dominant or recessive. There's no heterozygous possibility for males when we're talking about these sex-linked traits. And that's why you see the traits much more common in males than females, because females have to inherit two different alleles that both parents have to have the trait in order for a female to receive the trait. But males only need to get it from one of their parents. If the X chromosome they get from mom has it, they're going to get it. And so that means it's a whole lot easier for males to get it if their mom is a carrier. And if we go back, we can see that all the males who had it, their mother was a carrier. And we never see any females passing it on when a female is a carrier, because one comes from the mom, but the dad can only give that dominant X, and so that's going to win out. When we put that into a Punnett square, you want to show the sex chromosomes in addition to the trait. So kind of like we did with multiple alleles, you want to do that exponent notation. And so you're going to have XX for the mom, XY for the dad, and no alleles associated with that Y chromosome. And so you build it the same way, drop it down and slide it across, but we maintain those sex chromosomes because we need to know what the ratio is going to be for the males and what the ratio is going to be for the females. Because remember, one of those X comes from dad, one of those X comes from mom in females, but in males, since the dad gave the Y chromosome, the X only comes from mom. Okay, so this is going to be why these recessive X-linked traits are going to show up in males a lot more often. Only when it's recessive do you see this like more frequent ratio, and it'll be that 50% ratio when one of the moms is a carrier, for example, because you only need to get one of them, whereas the females have to get two. And there's it's a big chromosome, and so there's a lot of genes on it, and the Y has very few. And So this is why we're going to see more X-linked genes than we're going to see Y-linked genes. And usually when we're talking about sex-linked, we're talking about an X-linked gene. Another interesting form of inheritance is non-nuclear. So a great way to determine lineage for people who are really interested in pedigrees or ancestry is looking at mitochondrial DNA. So if you remember when we talked about cells, mitochondria and chloroplasts have their own sets of DNA because they originally were independent bacteria that were swallowed up. And so because of that, sperm are really small cells that are just trying to carry DNA to an egg. And so they don't have a whole bunch of extra organelles. And so you get all of your mitochondria from the egg given to you by your mom. And that means all of your mitochondrial DNA is the same as your mother's mitochondrial DNA. And so what that means is you actually have more information from your mom than your dad, because 100% of that mitochondrial DNA is inherited from your mother. And there's certain traits that could affect how your mitochondria perform. And so if your mom has that trait, all the children will. But if the dad has it, kids are lucky because it's only coming from the mom. And this was actually used in a really famous case. So in the Russian Tsars, the Romanov family, the last of the Tsars, were killed during the Russian Revolution. Now, there were a lot of rumors that some family members, like the youngest daughter, Anastasia, actually escaped. There was a whole movie about it that was made. Unfortunately, some remains were found, and using mitochondrial DNA to trace lineages of people known to be related to this family, they were able to identify that, yes, these were the Romanovs, and they were able to identify that Anastasia was among those remains. And so there were some people who claimed to be Anastasia, that she had escaped, and unfortunately they were able to prove that these were not true, but Anastasia had died with the rest of her family. Another type of trait, sex-limited, not sex-linked, but sex-limited, and these are traits that, like male pattern baldness, that are more dominant in one sex than the other. They appear more frequently. And then finally, I want to talk about linked genes. And as some of you have probably been starting to notice when we do multi-gene crosses, like dihybrid crosses, trihybrid crosses, you may say, like, well, will it always be random assortment? Wouldn't that only work if they're on separate chromosomes? What if they're on the same chromosome? Won't the certain pairings always be inherited the same? And that's true. Really, when we talk about independent assortment, we're talking about genes that are on different chromosomes. And if it's on the same chromosome, those traits are going to get inherited together. And so any genes that are on the same chromosome usually get inherited together. But there's one important step of meiosis that can interrupt gene linkage. Do you remember what that is? That's crossing over. Remember that those parts of chromosomes will trade with other homologous pairs. And so this can actually separate linked genes and make it so that the ratios we would expect of genes on different chromosomes can actually happen for genes that are on the same chromosome. But it depends on how far apart those genes are. If those genes are really close, the frequency that crossing over will happen in between those genes is really small. And so those are going to be linked genes. If the genes are on the same chromosome but super far apart, it's probable that a lot of crossing over events will mix these up. And so we may not get perfect 25% of each probability like in a Punnett square, but it'll be pretty close to those frequencies that it's inherited at. And if they're on different chromosomes, then they're not going to be linked at all. So let's talk about how this actually works. Let's break it down. So if two genes are on different chromosomes, like we're tracking how the different gametes we could make with gene A and gene B, if they're on different chromosomes, the combinations we have of AB, little a, big A, little b, is going to be 25% each because of independent assortment and meiosis. Now, Because of crossing over, if they're super far apart on the same chromosome, it's probably not going to be exactly 25%, but it's going to be really, really close. Remember, those chromosomes are going to trade parts on one of the chromosomes, and we're going to end up with roughly the same percentage breakdown. However, if they're really, really close, the recombination, or remember crossing over, another word for that is recombination, the amount that that's going to happen between those two is going to be really, really rare. It's not going to happen very often. Maybe that's going to happen here or here or here. It's not going to happen right in between those two letters. And so we'll see that we mostly get big A, big B, little b, and then the recombinant forms only 2% compared to 48%, whereas previously we had expected 25% of each of these genes. So when they're on the same chromosome, and they're really, really close, we can see that recombination won't always break them up. So can we look at data and figure out how far apart chromosomes are? You better believe we can. And so we can calculate how the recombination frequency, or how much we would expect these to reform. So we can see the parental traits of what we would expect to find based on genotypes, and then the recombinations would be those not present in the parents. And if we would expect, just based on pure independent assortment, random chance, we would expect each of these to be about a quarter. We can see there's a lot more of one than the other. We can see that these are the recombinant forms. And so what we're going to do is we're going to take all the recombinants in the offspring generation and then divide that by all the offspring in total. That's going to give us a decimal, multiply that by 100, and that's going to give us a percentage. So what can we do with that? Well, we can start to build a gene linkage map. So say we had 3 genes on the same chromosome, and we found out the recombination rate between A and B, between B and C, and A and C. We're going to assume that the largest recombination frequency are the furthest apart, the smallest is going to be closer together, and then we can start to figure out and build a map for how far apart these are. And we can use, instead of always mentioning the unit as percentage, we're going to use the unit of a map unit. So 1 percentage of recombination frequency is going to be the same as 1 map unit. So here where you see A to C is 18.5%, that you could also say is 18.5 map units. So you may be noticing, though, doing a little mental math and saying, these numbers don't add up, Mr. Malgren. Yeah, 13.2 plus 6.4 is 19.6, whereas that's a lot bigger, that's a whole map unit bigger than 18.5. How can this happen? Well, it's not always going to add up because, and it's not, that's why we don't use specific numbers, we use map units. We're not measuring in a specific measurement because double crossovers can happen. And so it's not going to tell us an exact distance measurement for us. There's lots of, you can have double crossover events happen on chromosomes, which gives us even more genetic variation, which in the long run is a very good thing. It may make it more difficult to calculate, but it's a good thing for our population. Last thing I want to talk about with crossing over events is sometimes rare events happen where crossing over doesn't occur between homologous chromosomes, but they'll be crossing over between 2 different chromosomes. So like 9 crosses over with 22. And this is going to be called translocation. Anytime crossing over happens between non-homologous chromosomes. And there's no set rule about how this could affect an organism. Sometimes it's silent or it has no effect. Sometimes maybe it creates a beneficial mutation, but it can also be detrimental. And there's a lot of things that have been associated with these translocation events, things such as leukemia, lymphoma. There's all sorts of events that can result in genetic diseases that we've associated with these translocation events. So anytime you're disrupting the DNA, it can potentially cause some problems. And the reason, because we're not crossing over with homologs, it's not the same break in the same place in the DNA, we could be interrupting genes on one or the other, and that can cause some serious problems for protein manufacturing. So that's our topic. That's our lesson today on chapter 12, big key take-home topics. You should be able to carry out a Punnett square and identify offspring showing codominance, incomplete dominance, multiple alleles, and sex linkage. If you can't do one of those Punnett squares, you need to do some practice. You should be able to understand that mitochondrial DNA is always inherited through the mother, and it's a way to trace genetic lineage. And then finally, you should be able to explain why certain alleles are inherited together more frequently than others. So looking at gene linkage, and then looking at those offspring, being able to build one of those gene maps. So if you're having trouble with any of those concepts, come see me in asynchronous. Otherwise, I hope you have a great rest of your day.